One of the most important and fundamental concepts in all of biology is the understanding that all cells require energy. Now, where this energy is coming from could be a multiple array of places. It may be coming from an organism that is consuming plants, such as a grazer, vegetarian of sorts. Sometimes it may be the situation where you have a predator, where an animal is consuming another animal. Or it could be a decomposer like a fungal species that is breaking down different waste materials or decaying tissue. Regardless of where they're getting their energy from, their cells do require energy. Now, the energy is going to be used for cellular processes. So we're talking about metabolism. We're also talking about reproduction. It could be movement. So there's lots of reasons why cells require a very high amount of energy, and it needs to be available at any given point throughout the day. Now, the basis of cellular energetics starts with how that energy is first synthesized. Notice that I'm not using the term created because that's not really accurate. It's actually a conversion process, and it all starts with the photoautotrophs. That is a really important word to know moving forward in this section. So this lecture is all about photosynthesis, the process, where it takes place, and what is made in the process. So let's start with the source of all the energy. Initially, all the energy is in the form of radiant light energy. These are photons of light. So the sun is pretty much the original source. What photoautotrophs do, and notice I'm not just saying plants, but plants are an example. Photoautotrophs are any organisms that capture sunlight using photosynthetic processes to convert into chemical energy in the form of carbohydrates and other macromolecules. So in this case, we use the example of plants. They're going to capture the sunlight and they're going to combine it with a couple other reactants to make the carbohydrates. So if you look at the chemical reaction, you have 6 carbon dioxide molecules, 6 water molecules with the combination of light. And then you have, in this case, it's solar energy from the sun. And it creates the products, glucose, which is C6H12O6, and 6 oxygen molecules. So products are on the right-hand side, reactants are on the left-hand side. It's good to just go ahead and memorize that, keep it as something that's understood because we're going to be revisiting these again shortly. So the 2 phases of photosynthesis, it's broken down into 2 steps or 2 stages. So we're going to start with the first one, light-dependent reactions, sometimes referred to as the LDRs. This is the first stage that takes place only when light is available. So in the case of, you think about the 2 parts of the day, day and night, the light-dependent reactions can't take place at night. The most important and critical component of the LDRs, in order for them to take place and begin, is the presence of light. So of course, they can happen during the day. Now, there are cases, artificial light and having, plants grow indoors, right? You provide the light, but that's kind of the first requirement for that. Now, the light-dependent reactions are basically a recharging station. It's where there are going to be a couple of very important molecules that are in low energy state that need to be re-energized, and we'll get to that shortly. The light-independent reactions, sometimes called the Calvin cycle, and I think often incorrectly sometimes called the dark reactions. For the sake of this video, we'll just call them the light-independent reactions or the Calvin cycle. Now, the reactants of the LIRs, or Calvin cycle, were the products of the LDRs. So we're going to talk about what things are processed, how they're recharged, where they go, and what happens to them. But do keep in mind there at the bottom, you definitely want to memorize the chemical equation of photosynthesis. So it's important. Before we move forward, it's important to understand that when it comes to photosynthetic activity, specifically when we're talking about plants, the photosynthetic region of the plant is going to be the leaf region. So leaves are basically organs for plants. Just like the human body has organs for certain functionality, the same can be said about plants. Now, there are some leafless plants, obviously, or they have modified types of leaves. But for the most part, that's the commonality. So this is where photosynthesis takes place. Specifically, the name of the organelle where photosynthesis takes place is the chloroplast. Now, the chloroplast is like a cell within a cell. And the reason it's considered this is because it has properties of what a cell has. So there's a double membrane. You could see in the picture there, an inner and outer membrane. It also has individual components within the chloroplast. So you have thylakoids, which are the little green discs, and then you have the granum, which are stacks of thylakoids. And that's the location of where you're going to see that the light-dependent reactions take place. Now the stroma is the fluid or gel-like substance that resides within the chloroplast. It serves to the same purpose of what the cytoplasm is to the cell. So a striking similarity, and this is why we say that the chloroplast is like a cell within a cell. Now the importance of the thylakoids is this is where the photosystems are located. Photosystems are collections of pigments. They are protein molecules that absorb sunlight. So the LDRs take place in the thylakoids. The stroma's importance is that this is where the Calvin cycle takes place. So this is where the second stage is going to occur. All the products from the LDRs are then taken to become the reactants in the LIRs. We'll talk more about that shortly. So what's the importance of chloroplasts and what they contain? Well, the chloroplast is really important because, remember, this is the site of photosynthetic activity. Now, the main molecules involved in absorbing sunlight are called pigments. The most important pigments that we'll talk about are the chlorophyll pigments, like chlorophyll A and B. They're able to do a lot of the primary light absorption for plants and photosynthetic organisms. There are some extra or additional different types of pigments, such as xanthophylls, which appear yellow, and carotenoids, which appear like orange and red. Those aren't primary pigments like chlorophyll A and B. Those are actually considered accessory pigments or secondary pigments. They pretty much absorb different types of light. So when you think about the spectrum of visible light, carotenoids are going to reflect orange and red because they are orange and red. So that's kind of the way it works. Whichever types of spectrum of light a pigment absorbs, that's the color it is not. It reflects the color it is. So that's kind of the way that works. So, chlorophyll A and B are really good at absorbing certain types of light, but carotenoids can do different types of light. So when a plant has multiple types of pigments, it enhances their ability to have more additional light absorption properties. Let's start with the LDRs, the first stage in photosynthesis. So remember, this is present in the thylakoid membrane, in the thylakoids. You have sunlight that strikes the chlorophyll molecules and maybe some other pigments like we just talked about on the last slide. The reason the sunlight is important in this case is the sunlight is going to be really critical in energizing the low-powered molecules that reside in the electron transport chain. So really it's going to be getting them excited, providing them with energy so that they become highly energized to be useful in the process. We'll also talk about what else sunlight is going to do. So to talk a little bit about the LDRs, you have to understand the importance of the electron transport chain. Residing in the thylakoid membrane, ADP, which is adenosine diphosphate, needs to become ATP. And NADP plus needs to become NADPH. And the way this works is that ADP is adenosine diphosphate. It needs to receive a third phosphate group, which is adenosine triphosphate. In ATP, that's a high energy molecule. ADP is a low energy molecule. So think about it like recharging a battery. And the same can be said about NADP+. NADP+ is an electron carrier, so it can take on the additional electron, go through the electron transport chain to process these molecules to use them in the Calvin cycle or the LIRs. So to think about it one way, since this is a cycle, the ADP, which is low energy, the NADP+, which is also low energy, they have to become energized. They're energized in the thylakoid membranes, in the electron transport chain, in the LDRs. Now, where are those two molecules coming from, the ADP and the NADP+? They're coming from the Calvin cycle where they were used, the LIRs. So they get recharged, they're regenerated to ATP and NADPH, so they can be reused in the LIRs, or in the Calvin cycle. So this talks a little bit about the LDRs. Probably the most important thing to note is the second note there. It says to replace the electrons that are used in the electron transport chain, the water molecule is split. This process is called photolysis. The water is required for this. So basically what happens if you look at the chemical equation, the chemical identification of water, H2O, what's needed and what happens is during photolysis, the water molecule H2O is actually split. The bonds between the hydrogens and the oxygen are severed. The H plus ions are retained. Using the electron transport chain, it creates an electromagnetic gradient, or a concentration gradient. The oxygens become waste. So when you look at the equation of photosynthesis, the oxygen is a byproduct. That's where the oxygen is formed. In the LDRs, when water is split by sun during photolysis, the H plus ions create this concentration gradient, allowing for the entering of CO2, the removal of O2. So we'll talk about more in a second. So this is pretty much the process here. Sunlight is used, absorbed by the chlorophyll and other pigments in the thylakoid membranes, splits the water molecule, oxygen is released, you can see that on the left-hand side. NADP plus becomes NADPH, readily usable in the Calvin cycle, and then you have the production of ATP and NADPH ready to be used in the Calvin cycle. So let's go to the second stage. Now, the light-independent reactions, or Calvin cycle, does not require sunlight. So in this case, quite different from the light-dependent reactions. So the reason why some people call this the dark reactions is because it doesn't require light. So this can actually happen at nighttime, contrary to the LDRs. So this takes place in the stroma, which is kind of like the cytoplasm of the cell. The stroma is basically the fluid inside of the chloroplast. This is where the products of the LDRs are going to be used. Now the O2 was already released, but CO2 is going to enter upon the release of O2. The structures involved are the stomata or the stoma of the leaves. So we talked about the guard cells, and we looked at our lab. We saw the openings. They allow for the exchange, the removal of the O2 and the entrance of the CO2. Now, upon the entrance of the CO2, the ATP and the NADPH from the light-dependent reactions can now be used in the Calvin cycle. So here's the process of the Calvin cycle. You now have your gas, which is available, the CO2. It's going to be used in combination of the ATP and the NADPH, the electron carrier. It's going to convert and change form a couple of times. So you'll see a couple molecules like PGA and PGAL, RUBP. I'm not going to get into any of those details, but at the end of this, the carbon dioxide that entered the stroma and the NADPH and the ATP from the light-dependent reactions combine together to form the glucose molecule, C6H12O6. And as long as the water molecules are being split, and as long as ATP and NADPH are generated, and as long as there is carbon dioxide gas, the Calvin cycle will continue to go through. So let's take a look and rehash everything from the equation. Carbon dioxide is used in the Calvin cycle. It's going to be entering through the stomata of the leaves through the guard cells, and in the process, they're releasing the O2 from the light-dependent reactions. The importance of the water is it's used in photolysis during the light-dependent reactions. The water molecule is split, the H plus ions are retained, the oxygen, the O2, is going to be dispelled. Sunlight is obviously important because it's required for photolysis. It actually does the splitting of the water molecule. And at the end of the Calvin cycle, C6H12O6, that is glucose, that is now being produced. And then, of course, plants can fix glucose into many different carbohydrates, lipids, and proteins. And the last thing mentioned here is the oxygen. Remember, the oxygen is produced in the light-dependent reactions and released as a byproduct. In order to exchange for the carbon dioxide used in the Calvin cycle, it must enter. So that's pretty much everything from the process aspect, but there are some things that you guys need to know. The first thing is there are many factors that can affect photosynthetic rates. There are 3 or 4 really important ones that I'm going to mention quickly. The amount of carbon dioxide that is present will affect how much of the Calvin cycle can take place. So remember, the stomata, the structures in the leaves, in the plant cells in the leaves, they have to open up in order to allow CO2 to enter and oxygen to leave. So without the stomata being open, plants are unable to retrieve and take in all the CO2. So it's really critical for the Calvin cycle that large amounts of CO2 are available. Now there is a negative trade-off, right? In this case, when plants open up their stomata, they're also going to lose some water in the process. So they're gaining CO2, they're letting out O2. That gas exchange needs to take place. But the unfortunate thing is they're also going to lose water to transevaporation or just basically evaporation into the atmosphere. So it's kind of a trade-off. You are giving up something to gain something, and it's something plants have to do. Obviously, this is a tougher thing in the desert. Plants are only going to open their stomata at night. We call that the CAM pathway. And then there's some other things like the C4 pathway, which does more photosynthetic activity with less CO2. The guard cells are only partially opening the stomata, so they can reduce the amount of evapotranspiration that occurs. The C3 pathway is the normal pathway. Most temperate areas don't really have issues with completely drying out and dying, so the management of the stomata is less of a concern. Now, along with those same conditions, temperature plays a big part. There's a certain optimal range for temperatures for plants and other photosynthetic organisms to make the process occur. If it's too cold, like say, sub 5 degrees Celsius, plants struggle to photosynthesize. The enzymes needed for this to take place, like ATP synthase and other enzymes, really struggle to work. Their effectiveness decreases as it gets too cold. So obviously 0 degrees Celsius, the enzymes aren't going to function well, and also water is going to be locked up as ice in that case. And the same thing goes for if it's too hot. Above 30 or 35 degrees Celsius, plants really lose their ability to photosynthesize well. Again, the enzyme functionality starts to decrease when it gets too warm. Another thing is the intensity and the amount of light. The more light, the better. Okay, think about it. You've got all this water available. Temperatures are ideal. If I have a lot of sunlight and I have a lot of photosystems and pigments for absorbing the sunlight, that's going to mean more photolysis, more splitting of the water molecule, which means more generated ATP and NADPH for the Calvin cycle. So obviously, the more intense the light, the more available energy there is. And the less available sunlight or light intensity, the less photosynthetic activity can take place. So that's all I have for you guys on this one. I hope you guys enjoyed it. Go ahead and rewatch, rewind, pause, take notes, look at different images so you can make sense out of the process. Next video coming up is going to be cellular respiration, which is part of the second part of the cellular energetics unit. So I hope you guys liked the video. I'll see you guys on the next one.